Hi, welcome everyone to this learning talk. My name is Shuaib Muhammad Jami from the University of Lagos, Nigeria. I'll be presenting on the histopathological alteration in some tissues of Heteroclarias exposed to Diclofenac with ameliorative potential of Neem Leaf as a director indica. The quest for quality life expectancy has increased the use of pharmaceuticals, both medical and veterinary importance, and as thus there's increment in the release of untreated and poorly treated sewage from pharmaceutical industry into water bodies around the world. And of all pharmaceutical drugs encountered in water bodies, non-steroidal anti-inflammatory drugs, as commonly called NSAIDs, has been reported to be found in water bodies, and due to the numbers of these drugs which are found on the global market, about 50 and more, including Diclofenac, ibuprofen, naproxen, among others. Have they report shown that these drugs are capable of inducing lesion and are capable of being a toxicant in aquatic organisms, particularly what Diclofenac. However, little to no attention is paid to the adverse effect of these drugs on living organisms, particularly aquatic habitat in the study area and other regions of the world. Moreover, laboratory studies on the use of medicinal plants as a potential ameliorative agent on the toxicological impacts of pharmaceuticals on fishes are scanty. As a result, these present studies aim to assess the histopathological lesion induced by lead and sublead concentration of Diclofenac in the gills, liver, and kidney of juvenile catfish Heteroclarias with an ameliorative potential of Neem Leaf. The juvenile of Abdi catfish used in this study was procured from a fish farm in Kwasu, Nigeria, after which the Diclofenac solution, which is the toxicant in this case, was prepared using 1 gram to 100 ml of 1 gram of Diclofenac to 100 ml of distilled water. After a successful age finding test, the fishes were exposed to acute exposure with varying concentration, including 4 mg, which is the lowest, and the highest, which is 20 mg per liter, to determine the 96 hours LC50 of the toxicant. Subsequently, the LC50 was obtained using the carbon arithmetic method, and the value is 10.8 mg, which is thus used in the sublead concentration for 30 days. The Neem Leaf, which is as a director indica, was obtained from the surrounding areas of the University of Lagos, and it was identified in the Department of Plant Biology of Faculty of Life Sciences in the same university and prepared according to Katisal et al. In 2008, which is thus included in the fish diet, respectively. At the end of the exposure period, the vital organ of the fishes, including the liver and the kidney, were obtained from the fish and preserved in 10% formalin bottle, which is then transported to the laboratory for analysis. Now, showing on the screen is the photomicrograph of the gills of the fish of the Heteroclarias exposed to Diclofenac for 96 hours, and the other one is for 30 days. Several lesions were observed in the gills from mild to severe, including the lamellar vision, epithelial lifting, rupture of epithelial cell, hemorrhage, and among others. Whereas the use of 1%, the inclusion of 1% Neem Leaf, as shown here, able to ameliorate the normal histology of the fish, of the gills. And also here is also the liver, the photomicrograph of the liver of Heteroclarias exposed to 90 hours and 30 days respectively. We have several the major histopathological lesions observed in the liver include the vacuolation, nuclear alteration, fatty degeneration, and necrosis. And similar cases as observed in the gills. 1% inclusion of Neem Leaf able to ameliorate the normal histology of the liver from others. With increase in use of Neem Leaf, that is 2%, and 4% turns out to be a potential toxicant. And lastly, this is the photomicrograph of the kidney exposed to 90 hours and 30 days respectively. We also have several lesions observed in the kidney, including disorganization of the cells, hypotrophy, and necrosis, among others. And the use of 1% Neem Leaf also able to ameliorate the liver, the kidney tissue, the kidney cell from the toxicant, that is Diclofenac. An increase in the use of the Neem Leaf, which is the ameliorative agent, turns out to be a potential toxicant. This is observed in the photomicrograph as shown on the screen here. And lastly, the study shown that the Diclofenac is a potential toxicant on fish with 1% inclusion of Neem Leaf able to ameliorate the normal histology of the tissue. This may, however, result in commercialization of the plant for large-scale